Good day. My name is Americane, and today I will be presenting on bioremediation in food systems. This focuses on the use of microorganisms and enzymes to remove or reduce toxic compounds in food and improve food safety. Bioremediation refers to the use of living organisms such as bacteria, fungi, or their enzymes to remove or neutralize harmful substances. In food systems, this is important because food can be contaminated with toxins such as mycotoxins and pesticide residues. The main mechanisms involved are biodegradation and adsorption. In biodegradation, microorganisms break down toxic compounds into less harmful substances. In adsorption, toxins bind to microbial cells, preventing absorption in the human body. The effectiveness of bioremediation depends on factors such as temperature, pH, moisture content, and the type of microorganism used. These factors influence how efficiently toxins are removed from food systems. Bioremediation is important in South Africa, where staple foods like maize are often contaminated. It provides a natural, sustainable, and cost-effective solution to improve food safety without using harmful chemicals. It is applied in plant-based foods such as maize, peanuts, and grains to reduce mycotoxins. In dairy products, microorganisms are used in fermentation to improve both safety and quality of the final product. Bioremediation is also used in beverages, especially fermented drinks, and in animal-based foods to reduce contamination during processing and storage, improving overall food safety. In practice, bioremediation is carried out through controlled fermentation or by adding specific microbial cultures. Environmental conditions such as temperature and pH must be carefully controlled. It is important that the microorganisms used are safe and non-pathogenic, and that the process does not negatively affect the nutritional quality of the food being treated. Food safety regulations, including South African standards and international guidelines, help ensure safe food production. However, there are still gaps in regulating the use of microorganisms for toxin removal. A key study investigated the removal of Aflatoxin B1 using lactic acid bacteria. Aflatoxin B1 is a highly toxic compound produced by fungi and commonly found in maize and peanuts. This toxin is linked to serious health risks such as liver damage and cancer. Reducing aflatoxin contamination is therefore very important for food safety, especially in developing regions. The study aimed to determine whether lactic acid bacteria could effectively bind and remove Aflatoxin B1. These bacteria are commonly used in food fermentation and are considered safe. Researchers tested 12 different bacterial strains, including Lactobacillus rhamnosus GG and LC705. These were exposed to contaminated solutions under controlled laboratory conditions. The bacteria were tested in different forms, including live, heat-treated, and acid-treated. This helped determine how treatment affects their ability to bind toxins. The study measured initial binding capacity and the stability of binding after repeated washing. This showed how much toxin was removed and how strongly it remained attached. Results showed that Lactobacillus rhamnosus GG removed about 78.9% of Aflatoxin B1, while LC705 removed about 76.5%. Other strains showed much lower removal. This shows that bioremediation is strain-specific, meaning some microorganisms are more effective than others in removing toxins from food systems. After washing, the amount of toxin retained decreased. GG retained about 49.5%, while treated bacteria retained up to 66 to 71%, showing improved stability. This suggests that treatment improves toxin binding by exposing more binding sites on the bacterial cell surface, making the process more effective. However, a limitation is that binding is reversible. This means toxins can be released under certain conditions, which reduces the effectiveness of complete detoxification. Bioremediation improves food safety without significantly affecting nutritional value. It may also influence flavor and texture, especially in fermented foods. From a consumer perspective, natural detoxification methods are generally accepted, although some concerns may exist about the use of microorganisms in food. In South Africa, bioremediation is highly relevant due to maize consumption and toxin risks. It provides a cost-effective and sustainable solution. In conclusion, bioremediation is an effective method for reducing toxic compounds in food using microorganisms. Further research is needed to improve its stability and application.